that started. Okay. All right. Now, I've got to get computer going. Sorry, we're running behind, but anyway. Here's the list. Now, I'm going to just chat a bit while we're getting things going. Remember last time, I think we had talked a little bit about muscles, right? And when it comes to muscle, remember there are three basic types. What are they? Right, skeletal, smooth, and cardiac. Where do you find smooth muscle? Yes, GI tract is one of them. And blood vessels, remember. And is smooth muscle voluntary or involuntary? Involuntary, right. What about cardiac muscle? Yes, well, cardiac muscle is only in the walls of the heart, and it's also involuntary, right? Skeletal muscle, on the other hand, is voluntary, largely. And that is the one we find, of course, it connects to the bones and ensures posture and movement. Now, when it comes to muscles, particularly skeletal muscles, they have coverings. And what is the covering of the entire muscle called? It's the E word, yes, but there's two E words.
I can't say it.
Epimysium, that's it, right. Okay, now, that covers the entire muscle. Now, what's it made of? Yeah, mostly dense fibers, connective tissue. There's going to be some elastic fibers in there too, because remember, it's going to have to stretch and recoil with the muscle. And then there are probably some reticular fibers as well. All right, so you've got a little bit of everything. Now, bundles of muscle cells are called fascicles. And a fascicle is surrounded by a connective tissue covering as well. What is that connective tissue covering called? Perimysium, right. All right, and remember now, the endomysium and the perimysium are all going to have blood vessels and lymph vessels and nerves in them, right? And then we get down to the individual muscle fibers or cells, and they're covered as well with something. What are they covered with? What's their covering called? Endomysium. And it's largely made of what kind of fibers? Reticular fibers, right. Now, all of these coverings extend beyond the muscle, okay? And what is that called when that extension goes beyond the muscle and connects to a bone? What's it called? What is that structure called? What connects a muscle to bone? A tendon, right. Okay, and so when muscle cells contract, they pull on all those coverings, you know, the epimysium, the perimysium, the endomysium, all those, and in turn that pulls on the tendon, which pulls on the bone, because the tendon, remember, is connected to the periosteum of a bone. All right, now, very simple, I think. Gets us to about where we want to start, because I think we want to start with attachments. And we should have it up here before too much longer. Slow as Christmas today. All right, there we go. There we go. So, there we go. Now, get the projector going. All right, attachments. Now remember there are two for any muscle. There's what's called an origin, and there's what's called an insertion. We have those terms in lab. What is the origin? What does that refer to? Less mobile part, right. The bone that either doesn't move at all or moves very little. And then there's the insertion, and that is, well, that's the bone that moves the most or does move. All right, so for example, if we consider the mandible and the masseter muscle, all right, that I was exercising a moment ago when I was chewing on my crackers. I don't have orange teeth as a result, but anyhow. But be that as it may, what is the origin for the masseter muscle? There's a structure right up in here. What's that called? Zygomatic arch. Okay, zygomatic arch. And it's, of course, parts from two bones, as you recall the formula. That's the origin. What's the insertion? The insertion would be the mandible, right? Okay, for chewing and grinding, right? Well, that's simple. Okay. Well, you all remember that, of course. All right, now, muscles can have either direct or indirect attachments. Well, that was an example of one that has a direct attachment, because the tendon is attached directly to the mandible. All right, so it causes the movement. But we can also have indirect attachments. Now here we're looking at attachment by way of things called, rather than necessarily the bones directly, but things called tendon sheaths or aponeuroses, which are like flat connective tissue sheets. And some examples there. Well now, there is a muscle that goes right across here, right? What is that muscle called? Well, bring back the E prefix. Means over. Say it louder. Epicranius. Right. And the epicranius has the two bodies or bellies, right? There's the frontal or frontalis part, and there's the occipitalis, right? Or occipital part. And then between the two is a flat sheet of connective tissue. All right? That would be like a tendon sheath that goes over the crown, if you will, of the skull. All right, down here we have this long, straight muscle. Remember what it's called? Remember, rectus means straight. Abdominis, like rectus abdominis, right? And it's attached to a couple of aponeuroses, flat sheets of connective tissue. Okay? All right. Well, there you go. That's indirect attachment. Now, what could be simpler than that? Not much of anything. Now, histology of muscle cells. I don't think we got to this yet, did we? Well, you'll enjoy it immensely. More terms to add to your growing lexicon. Now, muscle cells have some unique structures. So they need some unique names. And we'll start with the very first one. Muscle fiber just means a cell. So, you go to the cocktail party, cluster of people chattering about muscle cells, another cluster of people chattering about muscle fibers. They're all talking about the same thing. Now, what about? Well, we're talking about skeletal muscles. They vary considerably in width. 10 to 20 micrometers is probably more the average. But what about their length? Well, it depends on how long the muscle is. Because remember, from one end of that muscle to the other, that's the length of one cell. The cells run the entire length, for the most part, of that muscle. And so if the muscle is 25 centimeters long, you're going to have cells in there that are 25 centimeters long. Very big. Now, because they are so large, they're obviously going to have to be multi-nucleate cells, right? And they are, as we well recall from lab when we look at skeletal muscle cells. And they have a membrane around them, like any cell would have. Well, because that membrane is going to be somewhat modified over the usual quote-unquote garden variety cell, we're going to have a special name for it. And we're going to call it a sarcolemma. So the sarcolemma is, in essence, the cytoplasmic membrane of a skeletal muscle cell. Now, what's it made of? Well, like any cytoplasmic membrane, it's largely a phospholipid bilayer, right? And embedded in that phospholipid bilayer are proteins. But, as we shall see, there are some extensions of that membrane that pass down into the cell, and we're going to call them T-tubules, and don't worry, they'll be back to visit with us here in just a bit. Now, the cytoplasm is called the sarcoplasm. Why do we need a special name? Well, there are going to be some special organelles that we're not going to find anywhere else. There are also going to be some modified organelles that are different from those organelles in other types of cells. So, the sarcolemma is the membrane, the sarcoplasm is the cytoplasm. Alright, now, what are some of those specialized structures that are unique to skeletal muscle cells? Well, number one on the list, glycosomes. Now, we've had those in liver cells, or when you get to the liver in Biology 108, you'll certainly pick them up again. Glycosomes are granules within the cell that are there to store glycogen. Now, what is glycogen? That's something we've talked about before. What is it? What is glycogen? Well, we could classify it as a polysaccharide. What does that mean? Yes. But what about it? What's it made of? It's essentially many molecules of what specific monosaccharide? Glucose, linked together in a chain. And for glycogen, it's a branched chain. You know, we could also call glycogen starch. It is. I mean, you know, it's animal starch is another name for it. And it's a polymer, really, of repeating units of glucose in a branched chain. Where did it come from? Well, it came from putting together a bunch of units of glucose. Now, why would you ever do that? Why would the liver do that, I wonder? Well, you know, after you've had a meal, you've probably taken in an abundance of glucose, right, after you've digested and absorbed everything. More than you can use at that point in time. And are you wanting to do evaluations? Well,
This anatomy and physiology.
yes, you can have them. They're all kind of brain dead, I think. So I shall, go ahead. I'll edit that out of the recording. No, not the brain dead part. I'm leaving that partly around the hall. Crisp answers to these things now. Oh, let's see. Yeah, that's okay. They should be back in a minute. Mm-hmm. Well, that's quick. And they're also making a whole bunch of them in a row. Mm-hmm. Oh. These are fairly solid. These are different though than what we had before. Place, wasn't it cheese fries?
Dr. Griffin, I gave you my test last week. Is it in your office?
Yes, I haven't done anything with it yet. I've
Okay.
been lazy. I didn't do much of anything here for the weekend.
Good reason.
Well, if they're gonna let you go.
Anytime. In fact, it's probably in that folder there on the desk upstairs.
Yeah, I see a little one.
I just haven't done it. That's just lazy. That's all. I was working on that.
Did you start grading the one from yesterday yet?
No, that will be soon. The Wednesday. And let's see, for lab Wednesday, we'll be doing brains, and that is in lab. So you'll be brain. And then next Monday, we'll probably do brains and eyeballs together for the section. And that's also in the lab book, so.
What are we dissecting? Rats?
No, we have sheep brains, and we have, I think, cow eyes.
Okay.
Or sheep eyes, one of them. Can't remember which one we're from, but it's similar. I'm for sheep eyes, can't think of it. But anyway, but I know the sheep brains.
Problems with them, they're not submitting at all.
Probably just one.
I had a whole bunch of them submit and then these last few are.
The internet might have sucked down now. I'm gonna have to take call.
Doing those on purpose.
Yeah, they started a week or two earlier, I think, so I think they ran short of time. Let's see this one. I think it's a week earlier.
I put it on the page.
Also, because they can't see on the poster this time. Last time. Anyway. Let's see what's, I think it's 51-21 or something like that. Which doesn't make a lot of sense, because the other two are 51-18, 51-5-1-1-8, 5-1-1-9, 5-1-5-1-2-0. But anyway.
Who teaches microbiology?
Dr. Vargas.
I don't think I know who that is.
Well, he has two of the 107 labs. He had one this morning, he has one on Thursday.
Does he have short dark hair?
He's short.
Okay, I know who you're talking about.
His office is right over.
Okay,
he's a
yeah.
Dr. Chen.
Okay.
They're in the same office right now. soul. Couldn't we get the thing to work? Okay, there we are now. Yeah, this Wi-Fi down here is. Yeah, see? Oh, I think everybody's done now. Yeah. Okay, let's go ahead then. I think everybody's back now. All right, glycosomes. All right, now, as we said, they are vacuoles containing glycogen. Now, glycogen, of course, is made when there's plenty of glucose floating around in the blood. You've got extra glucose to store as glycogen. Now, we know the liver takes it up and then releases it later as glucose, but when you're between meals. Why would muscle cells want to have glycogen? Well, most of the energy it takes for muscle contraction, of course, is ATP. Where did we get ATP? The oxidation of glucose. What are those familiar pathways with oxidation of glucose? Glycolysis is one, and Krebs cycle is the other, right? Now, it makes a lot of sense, I think, that if you're baking a cake, you want to make sure you got plenty of sugar in the cupboard, because you don't want to have to run to the store at midnight to buy a bag of sugar, right? Well, the thing with the muscles are, they store this glycogen, so when they have to make a lot of energy by oxidizing glucose, they've got a ready supply of it already there on hand. And they don't have to wait for the blood to deliver every last molecule one at a time. Because that's going to take forever, it's not going to be very efficient. Myoglobin. Myoglobin is a heme-containing protein, very similar to hemoglobin. What does hemoglobin do for us? Bind oxygen, right? Bind and release oxygen. Well, what does myoglobin do? Exactly the same thing. Binds and releases oxygen. The only difference is, of course, where do you find it? Well, myoglobin isn't out in the blood, it's in muscle cells. Now, why do you suppose muscle cells want a hoard of oxygen? But now, how do they get their energy? Oxidation of glucose, right? Okay, and that thing shut off. Ah! Crap. Well, I tried. Anyway. Well, recording for some of you. All right. Oxidation of glucose. Now, which kind of oxidation of glucose is more energy efficient? Aerobic or anaerobic? Aerobic, with oxygen, right. Because how much ATP do you get per glucose when you're using aerobic conditions? 36. Glycolysis and the Krebs cycle. And oxidative phosphorylation all working. Take away the oxygen, how much are you going to get? What's the net gain of ATP per glucose then? 2. Because you're only getting it from glycolysis, and then you've got to use fermentation to keep glycolysis operating. Well, muscle can do that, but it's not nearly as energy efficient as the aerobic pathways. So it makes sense to keep that oxygen, keep lots of oxygen in the cell. You know, yeah, the blood can deliver oxygen, but it's not going to get there as fast. And when muscles are contracting and relaxing, they bulk up, and that impedes the flow of blood, which further makes it harder to get oxygen into the cell. So it works more efficiently if they've got their own cache of oxygen stored up. Myofibrils, those are fibers that extend the length of the cell. They can contract and they can relax. And so the ability of muscle to contract is due to those myofibrils. And we're going to examine them in a little more detail here in a bit. Sarcoplasmic reticulum. What does that sound like? Endoplasmic reticulum. Right. Now, we had two kinds of endoplasmic reticulum. What were they? Rough and smooth. All right, what was rough endoplasmic reticulum for? Proteins, right? What about smooth endoplasmic reticulum in other cells? Lipid synthesis and storage. All right, Well, the sarcoplasmic reticulum is a modified smooth endoplasmic reticulum, but it doesn't make anything. What it does is store and release calcium ions. Stores and releases calcium ions. Well, we're going to need calcium ions when we want cells to contract. It's necessary for muscle contraction. And then sarcoplasmic reticulum releases the calcium to get muscles to contract, and then it pulls it right back in and stores it when we want them to relax. T-tubules, those are going to turn out to be an extension of the sarcolemma, an extension of the membrane. And we've already mentioned that, and we'll keep harping on them as we go. All right, let's say a little more about myofibrils. Now, we said they run the length of the cell. And in fact, there are a lot of them in there. So much so that, as we know, they can account for up to 80% of the volume of the sarcoplasm. Volume of the sarcoplasm, largely myofibrils. Very important because they're going to cause the muscle to contract. All right? And they run the entire length of the cell. So how long would they be? Well, it depends on how long the cell is. If the cell is 25 centimeters long, you've got myofibrils in there that are 25 centimeters long. All right, so there we go. Now, the myofibrils are going to be related, of course, to the striations. And striations can be broken down into light and dark staining bands. All right? And as you recall with skeletal muscle, that was one of the hallmarks of skeletal muscle, were the striations. All right. The light staining bands are called I bands. The dark staining bands are called A bands. All right? And we need to remember A bands and I bands. All right? So, if they're light staining, they're I bands. If they're dark staining, they're A bands. All right? And you see these alternating light and dark areas when you look at a skeletal muscle cell. Now, within the A band, there are lighter, darker areas. We're not going to worry about these. I got carried away when I made up the outline. So, H zones, M lines, flush them. We're not going to deal with them, but we are going to deal with the Z disc. Now, the Z disc, or the Z line, goes down the middle of the I band, the light staining area. All right? And that Z disc is what defines a sarcomere. And what, pray tell, is that? Well, that's what we have right here in this nice $700 model that we bought several years ago, because it shows it so nicely. All right. The sarcomere is, in essence, the functional unit for muscle contraction. And so those myofibrils are nothing more than chains of these sarcomeres. One going, end to end, and then bundles of these end-to-end sarcomeres make up a myofibril. All right, and this is like the model for one sarcomere. All right, this red thing here on the end, that's a Z disc, that's a Z disc. So between two successive Z discs, that's a sarcomere. And the Z disc is down the middle of the I band. All right? So this is half of an I band here. There's half of an I band here. There'd be another one there and there, for the next sarcomere. All right? Now, there we are. So, here we have the sarcomere, and of course, when this contracts, what's going to happen? Those Z lines are going to get closer together, like that. And when it relaxes, they're going to separate like so. All right, so there we go. Sarcomeres are maybe about 2 micrometers long, and as we've already said, they're the functional unit of muscle contraction. Myofibrils are just chains of those, chains and bundles of them, if you will. All right, now, the filaments that make up the sarcomere. There are 2 kinds of filaments here. there are thick filaments and there are thin filaments. And as we note, each thick filament has 6 thin filaments surrounding it. All right? What about the thick filaments? Well, they're in the center, and they run the length of the A band, the dark band, which you go right here. And if I take this set off, here are a bunch of thick filaments. They're made of a protein called myosin. And myosin is in essence a fiber, and it has on the end of it a double head, these 2 heads. It looks something like a weird, and we'll see a picture here in a bit in the diagram, like a weird golf club with 2 heads on instead of 1. And there's a whole bunch of these bundled together, and that forms the thick filament. And the thick filament's right in the center of the A band, the dark staining A band. All right, and there we go. So made of myosin, and come on. And then the thin filaments, well, they're out here on the ends, and here's a whole bunch of thin filaments. And as you note, when we put this thing together, there's an overlap here of thick and thin filaments in the A band area. All right, here is one thin filament, and I'm going to pass this one around and you can take a look at it. Now, it's made of 3 different proteins. Well, actually, it's more than that, but 3 types of proteins. All right, and these are made of actin. And on the model, the actin will be these little red balls that you see kind of spiraling around the thing. That's actin. All right, and then there's another protein called troponin. That's the green balls that you see on here. And then the troponin, or tropomyosin rather, is the yellow filaments that spiral around the actin. So we'll pass that around. You can take a look and see how they're arranged. Mostly it's actin. That's the little red ball. And then there are a couple of molecules of tropomyosin. That's the yellow fiber that spirals around. And then the green things that are on there, which is actually made of 3 separate proteins, that's troponin or troponin. All right? And so those are the proteins that make up the thin filaments. All right? And as we already noted, each thick filament has 6 of those thin filaments surrounding it. Okay? Now, here is a diagram that kind of shows, if somebody doused the lights there in the back, this will show up better. Here's the muscle covered with its epimysium, and then here's a fascicle, number of fascicles, and then we pull out a cell. Now, these are myofilaments, so there are a number of those in here. Here they are within the cell. And these myofilaments are made up of course, the heavy and light filaments, the thick and thin filaments, the sarcomeres. And if we go on to the next diagram, here in essence would be a myofibril. And between the 2 successive Z lines, that is a sarcomere. Thick filaments, kind of the lavender, blue would be the thin filaments. There's the Z line, there's the Z line, so there you go. You notice how these are arranged in a chain. And they're bundled together to form a myofibril, all right? And it's the whole thing that's responsible for contraction, but the unit, the functional unit of contraction is in essence one of the sarcomeres, like what we have there in the model. All right, and here we have Z line, down the middle of the I band, the light staining I band. So from one Z line to the next one, that would be a sarcomere, all right? And here in the center of the A band, what is it? Well, it's thick filaments and some overlap with thin filaments when it's relaxed, like it is here, all right? And so there we go. So there's one sarcomere there, here's another one here. All right, and you can see how they're arranged in a chain. All right, the Z disc, as we said, is in the center of the I band. And the Z disc is, throw the lights back up. is composed of a protein, and that protein is connected. And that we probably should know as well. All right? And I think it makes perfect sense, because what does a Z disc do? It connects one sarcomere to another. So, connecting is an appropriate name for the protein. All right? So there we go. Now, okay, we don't need to worry about Desmond, but now we need to worry about this one. The elastic filament. Now, where are we going to find that? Well, it doesn't really show up on this model, but we can show you where it goes. The elastic filament runs from the end of a thick filament over to the Z line. So it really attaches the end of the thick filament to the Z disc. And it is also composed of a protein, and that protein is called titin. And it's kind of like a spring, in a sense, because it really has a couple of functions. One thing that titin does is it resets the sarcomere when the muscles relax. Now, here's what happens. Now, when it contracts, the thin filaments overlap the thick, like that. When it relaxes, titin pulls the Z discs back to their relaxed position. So it's like a spring. All right? Compress it this way, and then it relaxes, and it goes right back to that reset position, back to the relaxed position. Another thing that the elastic fibers do, they prevent the overstretching of muscle. Because remember, that was another property of muscle. It can be stretched. And when you stretch it, what does it do? It recoils back to its original length and shape. Well, if I'm stretching it, what am I doing? I'm pulling these Z discs further apart. Titin, or the elastic filament, will limit how far I can move them apart. All right? So it helps to prevent the overstretching of muscle, as well as, being responsible for the relaxation of the sarcomere when the muscle relaxes. All right, so there we go. All right, and so here we are with another diagram, and I like this one because there's a electron micrograph, and here we have a drawing made from it, and then here we have another diagram that actually shows the filaments. So we have the thick filaments, the thin ones. That is, of course, the elastic filament, which is made of titin. Here's the Z line. What's the Z line made of? What was the protein that makes it up? Connectin, right? Connects one sarcomere with another. Okay. And the thick filaments were made of myosin, right? And the thin filaments, which are these light blue lines here, those are made of three types of proteins. What are they? Actin, troponin, and tropomyosin. Right. Okay? Now there we are. Now we got the table set with all the goodies. All right. Myosin thick filaments, as we said, rod-like with two globular heads on the end. What are the globular heads going to do? When the time comes, they're going to go up, they're going to grab onto actin. Now, where's the actin? Thin filaments, right? Okay. Now, the thin filaments, as we said, actin, globular protein, on the actin molecules, there is the site for myosin head binding. When the muscle is relaxed, that binding site on actin is covered up by tropomyosin. All right? When the muscle is contracting, the myosin binding site on actin is exposed. All right? It's not covered up. All right? So tropomyosin, as we've mentioned, these two spiral strands you saw going around that thin filament, the yellow-like threads, when it's relaxed, those are actually covering up the myosin binding sites on actin. When muscle is contracting, the tropomyosin has moved off and exposed those myosin binding sites on actin, so the myosin heads can attach. But what caused it to move? Oh, the other protein, troponin. Troponin is made of 3 proteins, one of which binds calcium ions. Calcium ions are going to be key here. Because calcium ions come in, they bind to troponin. What does that do to the shape of troponin? Changes it. The troponin then pulls the tropomyosin off of the myosin binding sites on actin and exposes them. And now myosin heads can bind. And that will cause contraction of the sarcomere, and thus contraction of the myofibril, and thus contraction of the muscle. Now, if you want to get muscle to relax, what do you think you're going to do with the calcium ions? Get rid of them. Alright, so you take the calcium off the troponin, it goes back to its original shape. And as it does, it pulls the tropomyosin back over the myosin binding sites on actin. Where the myosin head's going to attach? Nowhere, they can't. And tighten, springs the Z discs back to their original relaxed position. Simple, isn't it? Yes. Don't worry, we'll harp on it enough times you'll be able to see it in your sleep. Now, I love this diagram because it makes use of food images. Alright, to start with, here is a thick filament. What's it made of? Myosin, right? Myosin is this long strand-like thing with these 2 globular heads on the end. It looks sort of like a weird golf club, right? 2 heads on it instead of 1. Alright, there they are. Projecting out away from this band, and it is a band of myosin molecules. There's a whole bunch of them there. Now, here are the thin filaments. Now, what are they made of? Well, they're made of actin, mostly. Actin's a little globular protein. Now, here's where the food images come in. This probably doesn't show up terribly well, but they look a lot to me like little purple pimento stuffed olives. The pimento is the myosin binding site. Alright? But it's covered up. What's covering it up? Well, these strands of blue spaghetti are covering it up. Right? Okay. And what's attached to that blue spaghetti but little blue croissant? Don't they look like croissant? Alright. That is troponin or troponin. 3 proteins. Alright? Now, calcium comes in and binds to one of these. That changes the shape. And you notice this troponin is actually attached to the tropomyosin. So when it changes shape, it pulls that off to the side and exposes the myosin binding site on the actin. Now, the heads of myosin have a place to attach. Alright? And that's what's going to happen when we get ready for muscles to contract. Take away the calcium, goes right back to this conformation. There we have the troponin and the tropomyosin now covering up those myosin binding sites on actin. Nowhere now for the myosin heads to bind, so tighten pulls the Z discs right back to their resting position. And there it is. Alright, that's simple. Okay. Now a bit more about the sarcoplasmic reticulum. As we said, it's a modified smooth endoplasmic reticulum. And here it is on our model. Alright, you see the green stuff here, right? Those are, in essence, the smooth endoplasmic reticulum, sarcoplasmic reticulum as we call it here. And when this is relaxed, that sarcoplasmic reticulum is filled with calcium ions. All right? Now, the terminal cisternae, that line right there and there, those are terminal cisternae of a sarcoplasmic reticulum. Here's another set over here. And what have we got running between them? Well, that yellow line between the terminal cisternae is a T-tubule. And where do we get the T-tubule? It's an extension of the membrane of the sarcolemma. All right? But we're not ready for it just yet. As we've said, the major role for the sarcoplasmic reticulum is to store and release calcium. All right? It stores calcium when muscles relax. It releases calcium when you want muscle to contract. And when you want it then to relax again, that calcium is going to get pumped right back into and stored in that sarcoplasmic reticulum. All right? Now that's pretty simple, isn't it? Now where do you find these? Well, they're wrapped around every one of those myofibrils. And if we just for a second go back to this model, there's a myofibril. All right, here are parts of sarcoplasmic reticulum. There's the terminal cisternae here and here. And that whitish thing between the two, that's a T-tubule, which is an extension of the membrane of the sarcolemma. All right, so those sarcoplasmic reticula are, in essence, running around or surrounding, of course, each of the myofibrils. And the T-tubules, we said, that runs between the terminal cisternae of the sarcoplasmic reticulum. And it is an extension of the membrane or the sarcolemma. Now, why do we need it? Well, this is how we're going to spread the impulse down into the sarcoplasm to get these things to all contract together. All right, so let's take a look at this diagram. And so here we are. Here is the membrane, the sarcolemma. All right, and each of these little holes that you see here, those are the openings of a T-tubule. And here is the T-tubule extending down into the sarcoplasm. And on either side of the T-tubule is the terminal cisternae of the sarcoplasmic reticulum. Here's sarcoplasmic reticulum in between these, of course. And the sarcoplasmic reticula are wrapped around each of those myofibrils. So, if we have then a depolarization or a change in charge spreading across this membrane, it also spreads right down in here, as we shall see in a moment. All right? Now that's simple enough, isn't it? Oh, don't worry. We'll keep harping on it. Now, how do we get muscles to contract? Well, if we're talking about skeletal muscles, we have to supply nervous stimulation. And that is going to involve a neuromuscular junction. Now, what on earth is that? Well, if you get out your terminological dissecting kit, neuromuscular sounds like nerve connecting with muscle, doesn't it? Well, that's exactly what it is. All right? Each skeletal muscle cell is connected to a nerve fiber. Every one of them. Each one of them. All right, now that's not be true with the other types of muscle, but it is for skeletal muscle. Every fiber has a nervous connection. All right? Well, that can be important if you want to get graduated contraction. I mean, like, you know, I've got to have muscles here, you know, contracting to grip this thing. Well, I don't want every single muscle fiber in here to contract at once. What would happen to my remote control if it did? I'd probably crush the thing, right? Well, I don't want to do that. I want it to work. All right, so I don't necessarily want every cell contracting at once. I just want some of it. Well, if I can control each one individually, I can do that. If they all contract together, well, it's, that's all you can do. Well, you don't want to do that. It's not very useful. All right, neuromuscular junction. Site where a nerve and a muscle connect. Now, some other terms to add to your growing lexicon. Motor end plate. Well, that is actually on the muscle cell. What is it? The motor end plate is a modified sarcolemma, modified membrane on the muscle, where the nerve connection occurs. How's it going to look? Well, it's going to be folded up. There are going to be folds or pleats in that membrane at that location. That folded up or pleated up membrane is what's called the motor end plate. Now, a motor unit, well, that's the combination of the motor neuron that controls the muscle and the muscle cell itself. All right? So, the nerve that controls the muscle and the muscle together, that's a motor unit. Well, they operate as a unit. Impulse comes down the nerve. What's it going to do? It's going to cause the muscle to contract. And then we have another thing, a synaptic cleft. Well, it's a gap, if you will, a gap between the end of the nerve and the motor end plate of the muscle. There's a space between the two. Nothing in that space normally. All right? What is it for? Well, when we want to get the muscle to contract, an impulse comes down the nerve is going to release a transmitter substance into that cleft, into that gap. And that neurotransmitter goes across the gap and combines with receptors in the motor end plate area. All right? Now, there's one more wrinkle to the prune that isn't on the slide, so we'll just write it on the board. And that is something called a synaptic knob. Synaptic knob. Now, where are we going to find that? Well, that is at the end of the nerve. And it's kind of a little knob-shaped ending, if you will, at the end of the nerve, where the neurotransmitter substance is stored and released. The substance that will cause the muscle to contract is stored and released out of that synaptic knob at the end of the nerve. Now, we have a nice diagram that shows all this stuff. So let's get to it. All right, here we go. All right, here's our muscle cell. Here's the membrane. There's the folded up membrane. Now, what was that folded up membrane called on the sarcolemma? The motor end plate. All right. Now, what have we got here? Here is the terminal end of our nerve. It's the axon of the motor neuron. And you notice it branches out, and at the end of each of those branches is a synaptic knob. Looks like a knob, doesn't it? All right, so in yellow, that's nerve, and then in this beige coloring, that's the muscle cell. All right, up here we have it enlarged. There's a synaptic knob, all right, and here is the motor end plate. And then that gap between the two, the motor end plate and the synaptic knob, that's the synaptic cleft. All right? Remember that one? Now, an impulse comes down this nerve and it reaches that synaptic knob. And what that's going to do is going to cause these synaptic vesicles in here to lyse. Now, what's in those synaptic vesicles? A neurotransmitter substance. What's the neurotransmitter substance going to do? it's going to come right into that synaptic cleft. And it's going to go right over, and it's going to bump into that membrane, and embedded in that membrane are going to be receptors for it. And so we get a specific combining of the neurotransmitter substance with its receptors. And the receptors are embedded in the motor end plate of the muscle cell. All right? Now, what is the neurotransmitter substance? Well, let's take a look. Cut the lights, if you would, please, and we'll take a look at it. This is an actual photograph of one of these neuromuscular junctions. Here's the cell, muscle cell. There's the axon. Here are all those synaptic knobs. All right? And each one of these now would be releasing that neurotransmitter if an impulse comes along here. All right. And this is just showing how one nerve might cause several muscle cells to contract together. All right? And they do. But not every cell in that muscle is going to contract, at the same time, unless you want it to. All right. Now, what about the impulse for contraction? Well, here we go. Okay, we put the lights back on now. All right. And since I'm getting tired here, I'll just sit and yak. Because I can see I've already got your faces showing me that you're deer in the headlight. Look, but that's all right. I'm not bad. All right, now, what is the neurotransmitter substance? Well, it's acetylcholine. Now, that's a pretty name, isn't it? You might as well get used to acetylcholine, because when we get to the nervous system, we're going to use it over and over and over again. It's probably the most commonly used neurotransmitter substance in the body. Okay? It's a chemical substance. And it is typically released from nerve cells, neurons. All right? Now, impulse comes down the neuron, acetylcholine gets released. What does it do in the case of the muscle? It diffuses across that synaptic cleft and combines with acetylcholine receptors, which are proteins embedded in the membrane of the muscle cell at that motor end plate location. Okay? All right. So, if you want the muscle to contract, you've got to get the nerve to release the acetylcholine. All right? That's simple enough. Now, acetylcholine goes across the synaptic cleft, combines with the acetylcholine receptors, which are transmembrane proteins you find embedded in the membrane at that motor end plate location. All right? Everybody with us so far? Now, what happens next? Well, the motor end plate, the membrane, that motor end plate area, will now depolarize. Oh, what does that mean? Well, membranes are typically polarized. They have a charge on them. And the net charge on polarized membranes is negative, has a negative charge. All right? Now, what is going to happen when you do that? When you depolarize? Well, if it's polarized as a negative charge, it becomes depolarized, it takes on a positive charge, right? Yes! Now, how did it get from a negative charge on the surface to a positive charge? Well, sodium ion channels that are in that membrane. Now, what would a sodium ion channel be? Probably a transmembrane protein that lets what go through? Sodium ions, right? Yes. When it gets opened up, sodium ions go flooding through it into the cell. Now guess what happens to the charge on the membrane at that site when you open up the gates and sodium ions just go flooding right in, willy-nilly. It depolarizes. What does that mean? What happens to the charge? It was negative to start with, now it's positive, isn't it? Because what's the charge on a sodium ion? +1. Yes. Alright, so that section of the membrane has depolarized. It has changed charge. It's changed magnitude. It went from being negative to positive. Alright? Just in that one spot. Well, that's only temporary. Because now, guess what's going to happen? Well, there are sodium ion channels embedded in that membrane its entire length. And so, when the sodium channels in one area open, they cause the sodium channels in adjacent areas to open up too. So what happens? You get propagation. You get depolarization occurring the entire length of that muscle cell. The whole thing. The entire length. Alright? Now, does it just stay there on the surface? No! Because what are those extensions of the membrane called that go right down into the sarcoplasm? What are those things called? Starts with a T. T-tubules, right. And guess what? They've got sodium ion channels embedded in their length as well. So guess what's going to happen? Depolarization comes to one of those T-tubule holes and it falls right in. And it spreads right down the T-tubule into the sarcoplasm. Now, get the diagram. There in yellow, there are the T-tubules. What butts up against the T-tubules? What's the green stuff? Terminal cisternae of the sarcoplasmic reticulum. That's right. So if these T-tubules are depolarizing, guess what they're going to cause to happen in the terminal cisternae and the rest of those sarcoplasmic reticula? They're going to cause calcium channel ions in these things to open up. And calcium ions are now going to flood into the sarcoplasm. And when those calcium ions flood into the sarcoplasm, what are they going to bind with? Troponin. And when that happens, the troponin changes shape. It pulls the tropomyosin off of the myosin binding sites of actin. And once that's done, those myosin binding sites on actin are now exposed to the myosin heads, and there you go. You've got muscle contraction. Now, isn't that neat? Now, that's a lot of stuff gang together. But let's dissect a little bit more, and we'll pick up with it again next time. All right, so the acetylcholine goes across the synaptic cleft. It binds to acetylcholine receptors in the motor end plate of the muscle. What happens to the membrane in that sector of the muscle? It depolarizes. What did we say depolarization meant? Changing what? The charge goes from negative to positive. Why? Because in that area sodium ion channels popped open and let sodium in and did it. Alright, now, that area depolarizes first, but what happens with that depolarization business? It propagates. It spreads to all the adjacent areas on the surface of the cell and down the T-tubules into the sarcoplasm. And once it reaches the terminal cisternae of the sarcoplasmic reticulum, what happens there? The calcium channel open up in the sarcoplasmic reticulum, and calcium floods out into the sarcoplasm, and the muscles contract. Now, if you want the muscle to relax, what are you going to have to do? Well, the first thing you're going to have to do is get rid of acetylcholine. But that's easy. That's easy. If you want the muscles to relax, you've got to get rid of that acetylcholine. Well, there's an easy way to do it. Embedded in the membrane of that motor end plate, there's another protein. Now, there were acetylcholine receptors there, but there's another protein right beside it. It's called acetylcholine esterase. And acetylcholine esterase is an enzyme. And what does it do? Well, it basically neutralizes acetylcholine. It turns it in to acetic acid and the amino alcohol choline. Breaks it down, in other words. Neutralizes it. Now, what did acetylcholine do to that motor end plate originally? When it got over there and bound to its receptors, what did that do? What did that cause to happen to that area? It caused it to open up the sodium channels and depolarize. That's right. Now, if you get rid of the acetylcholine, what do you suppose is going to happen to the sodium ion channels? They snap closed. And the sodium now gets pumped right out. And what happens to the membrane? It repolarizes. It goes back to its resting potential. Negative charge, right? Well, now what's going to happen to the adjacent areas of that membrane? They're going to repolarize as well. What's going to happen down in the T-tubules? They're going to repolarize. And now, if those T-tubules, bless you, have repolarized, what's going to happen here with this sarcoplasmic reticulum stuff? It's going to pump that calcium right back in where it came from. There's going to be a calcium ion pump here that's going to pump the calcium right back into that sarcoplasmic reticulum. And when you pull it out of here, what do you suppose is going to happen to this sarcomere? It's going to relax. That's right. Because now you've taken the calcium off the troponin. It goes back to its original shape, pulls the tropomyosin back over the myosin binding sites on actin, and tightens, pulls those Z discs right back to their resting position, and the muscle relaxes. Now, isn't that simple? Heavens, yes. Now, one more little wrinkle, and then we'll call it a day, because I can see you're all glazed over. But that's all right. That'll make you study that much the more before Thursday. Nerve gas. There are lots of different ones. Nerve gas, okay, you've heard of those. Now, what do they do to you? What is a nerve gas?
What'd you say about Thursday?
Well, we'll go over all this stuff, and you'll be ready to pick up where we left off on Thursday. Oh, we have more to add. All right, but, well, don't worry about it. What does a nerve gas typically do to you? Kills you, right, yeah. Well, there are several different ones, but one of them, some of the more infamous ones, inhibit the action of this enzyme. They basically denature that enzyme. They interfere with its activity. So what happens? Well, Nerves get stimulated, release acetylcholine, muscle cells contract. If you don't have acetylcholine esterase there to inactivate the acetylcholine, what happens to the muscle cell? Exactly. Acetylcholine comes along. No acetylcholine esterase, no relaxation. Diaphragm contracts, stays contracted, you suffocate. Neat. Now, there's another case where you're also going to get that contraction, it doesn't relax. And it really doesn't have anything to do with acetylcholine esterase in this case. You've heard of rigor mortis? What's that? Muscles get rigid when you croak, right? Well, as we'll find out, when you die, you stop making ATP. You gotta have ATP to pump the calcium back in here. It escapes, it doesn't get pumped back in, the muscles just stay contracted until the proteins start to break down, and then you'll relax. As you start rotting away, of course. But anyway, that's another issue. All right, now there you go. Now isn't that simple? Isn't it? Now that depolarization, repolarization membranes, we're going to see that again when we talk about nerve conduction. Because it works in much the same way. It's moving ions in and out of a cell. And that alters the charge on the cell. And really when it comes to nerve transmission, just like with muscle contraction, it's an electrochemical process. Now isn't that simple? Isn't that neat? Oh, heavens, yes. Well, you've suffered enough for today, I can tell. But that's all right. We'll add to the lymph, well, next time.